Welcome back, everyone, to the Deep Dive. We've got a ton of breaking news in bioinformatics today, so let's jump right in.
Always exciting to see what's new in this rapidly evolving field.
Absolutely. First up, we're diving into the always fascinating topic of aging. A new study from UC San Diego, published in Nature, aging may have just cracked the code on why we age, connecting the dots between the two leading theories.
Yeah, for a long time, genetic mutations and epigenetic changes were considered separate explanations for aging.
Right, like two sides of the same coin.
Exactly, but this study suggests that those random mutations in our DNA can actually trigger specific epigenetic changes, which then contribute to the aging process itself.
So it's not just our genes, but how their expression changes over time that really matters.
Precisely. Think about the implications for anti-aging research.
It's pretty mind-blowing. Imagine being able to target those specific epigenetic changes to slow down or even reverse aging. That would be a game
Wow.
changer.
That would be revolutionary. Speaking of groundbreaking research, let's move on to the Human Pangenome Reference Sequencing Project, which just received a significant boost in funding from the NIH, a $14 million grant to be exact.
That's a substantial investment. What's the big picture goal of this project?
Well, the goal is to create a much more comprehensive and diverse reference genome for humans.
So not just based on a single individual like the current reference genome.
Exactly. The current one doesn't reflect the incredible genetic diversity of humanity. This project aims to create a more inclusive genetic mosaic that truly represents us all.
That makes a lot of sense. A more representative reference genome would have huge implications for both research and medicine, right?
Absolutely. More accurate diagnoses, more effective treatments, and a better understanding of how different populations respond to therapies. It's a big step towards personalized and equitable healthcare.
It's exciting to see how this project will unfold. But for now, let's shift gears and zoom in on a specific disease: breast cancer. We all know how frustrating it can be to get inconsistent results from biomarker tests, which can make treatment decisions so much harder.
Those inconsistencies can be a real roadblock to effective treatment. But there's some promising news on that front. Researchers at Comenius University Science Park are developing a new tool called MFIS-8-seq. It could offer much more precise and reliable information about tumors.
That sounds promising.
Mm.
What makes this tool different from existing biomarker tests?
Well, MFIS-8-seq combines two powerful techniques: fluorescence in situ hybridization and RNA sequencing.
So it's like a two-pronged approach.
Right. This allows researchers to identify very specific genetic biomarkers and actually see their precise location within the tumor tissue.
So it's not just about identifying the genes, but also understanding their spatial arrangement within the tumor. That sounds like a major step forward.
It is. That spatial information could explain why some tumors respond differently to treatment, even if they share similar biomarkers. MFIS-seq could lead to more targeted therapies, minimizing side effects and improving outcomes.
Now, get ready for a rapid-fire round because we've got a bunch of exciting breakthroughs in the field of single-cell genomics to cover. It seems like this field is exploding with new discoveries.
It really is. Single-cell genomics is transforming how we understand biological processes at the most fundamental level.
First up, we have Chronicell, a new software that's changing how scientists analyze single-cell data. It allows them to analyze data over time.
It's like watching a movie of cellular processes instead of looking at static snapshots.
Exactly. Imagine being able to track how individual cells change and interact over time.
It's a powerful way to visualize dynamic processes like cell differentiation, development, and disease progression. By tracking how gene expression changes in individual cells over time, we gain a much deeper understanding of how these processes actually work.
It's incredible to think that we can now observe these microscopic events unfolding in real time. And speaking of uncovering hidden details, direct RNA sequencing is shining a light on the world of rare RNA editing events.
RNA editing is a fascinating process that can change the sequence of RNA molecules after they've been transcribed from DNA. These changes are often subtle, but can have a big impact on gene expression.
So we're not just looking at the genes themselves, but also how their messages are being modified after they've been transcribed.
Precisely. And understanding these RNA editing events could be key to developing new treatments for a wide range of diseases. Some cancers, for instance, have been linked to abnormal RNA editing patterns.
That makes sense. It seems like with each new discovery, we're uncovering even more complexity within the world of single-cell genomics.
And with that complexity comes a lot of data. Analyzing massive amounts of single-cell data can be a real challenge, which is why a new R package called STRIA is so exciting.
This package makes it easier for researchers to analyze and compare data from multiple single-cell experiments.
It's all about streamlining the process, leading to faster insights and fostering collaboration among research teams.
Collaboration and data sharing are crucial for accelerating scientific progress. Speaking of speed, an enhanced sequencing technique called SmartSeq plus 5 Fun is helping us understand the very early stages of mammalian development.
This technique is giving us unprecedented insights into how an embryo's own genes take control from maternal influences during those critical early stages of development.
It's like witnessing the very essence of life unfolding at the molecular level.
And we're not just deciphering which genes are expressed, but also how efficiently those genes are translated into proteins. New sequencing technologies are enabling us to measure translation in single cells, adding another layer of complexity to our understanding of cellular function.
It's like going from reading a blueprint to watching the actual building being constructed. All of these single-cell genomics advancements are pretty remarkable.
They really are. It's an exciting time to be working in this field. The possibilities seem endless.
So what stands out to you the most from all these single-cell genomics breakthroughs?
I think the potential of Chronotel is particularly intriguing. Being able to observe dynamic cellular processes unfold over time is a game changer. It's like having a front row seat to the microscopic world.
It is pretty remarkable how far we've come in our ability to study and understand individual cells.
Uh-huh.
But let's move on now to another exciting frontier: artificial intelligence. Researchers are harnessing the power of AI to tackle some of the biggest challenges in healthcare, like Alzheimer's disease and cancer.
AI's potential in healthcare is truly transformative. It's changing how we diagnose, treat, and even prevent disease.
Let's start with what sounds like a potential breakthrough in Alzheimer's treatment: the use of radio frequencies.
It does sound futuristic, but researchers are exploring how radio waves might be used to target and potentially even treat Alzheimer's. It might sound like something out of a sci-fi movie.
Right. Using radio waves to treat Alzheimer's. How did researchers even stumble upon this idea?
It was actually a serendipitous discovery about 15 years ago. Researchers were studying a mouse model of Alzheimer's, and they happened to notice that exposure to radio frequency energy seemed to improve cognitive function in the mice.
So it's kind of an accidental finding.
It was. And while it's still very early in the research process, there's growing evidence that this approach, which is non-invasive, by the way, could help clear those amyloid plaques that are a hallmark of Alzheimer's.
So we could be moving beyond just imaging the brain to actually using these radio frequencies to modify brain activity.
And potentially even reverse the disease process. It's a very exciting possibility, but of course, more research is needed to fully understand the long-term effects and safety.
Absolutely. It's definitely something to watch closely, though. Now let's turn our attention to another area where AI is making waves: predicting cancer treatment response. Mount Sinai has developed a tool called Scorpio that uses AI to analyze blood tests and clinical data to predict how well patients will respond to immune checkpoint inhibitor therapy.
Immune checkpoint inhibitors are a really remarkable class of cancer drugs, but they don't work for everyone. Being able to predict who's most likely to benefit would be huge.
It would be life-saving for some patients and would also spare others from going through treatments that might not be effective for them.
Exactly. Scorpio could help doctors make more informed treatment decisions, leading to a more personalized approach to cancer care.
So it's all about using data to tailor treatment to the individual patient.
That's the goal. And what's especially interesting about Scorpio is that it doesn't rely on complex genetic testing.
No.
It uses data that's readily available from routine blood work, making it a potentially accessible tool for a wider range of patients.
That accessibility is so important when it comes to new technologies. We've covered so much ground today, from aging to the human pangenome to single-cell genomics and now AI. As we wrap up, is there an overarching theme or takeaway that you see emerging from all these breakthroughs?
I think the common thread is that bioinformatics is no longer just about describing biological systems. We're moving into an era where we can understand, predict, and even manipulate those systems in ways we never could before.
It's like we're shifting from being passive observers to becoming active participants in the story of life.
Precisely. And that's incredibly exciting, but it also comes with a tremendous responsibility to ensure that these powerful tools are used ethically and for the benefit of all humankind.
It really is a remarkable shift. It makes you think about the ethical implications, too. As we wrap up our deep dive into these bioinformatics breakthroughs, what's the one key takeaway you'd want to leave our listeners with today?
Bioinformatics is no longer some niche field tucked away in labs. It's front and center in medicine, healthcare, and really our understanding of what it means to be human.
It's shaping the future, and it sounds like the pace of discovery is only accelerating.
It definitely is. The potential for good is immense, but we need to keep those ethical considerations in mind as these technologies continue to evolve.
Absolutely. We have to ensure they're used responsibly for the benefit of everyone. So for our listeners out there, if you're as fascinated by all this as we are, keep exploring these topics.
Definitely don't stop here. Keep asking questions and stay informed about the incredible advances that are happening all the time in bioinformatics.
It's a field that's changing the world as we know it. Thanks for joining us on this deep dive into the latest bioinformatics breakthroughs.
It's been a pleasure. Until next time.
See you then.